Hi everyone, I'm going to talk you through the process of genetic engineering, which comes up in Biology Paper 2 for combined and triple science students. And I've produced a diagram that I'm going to refer to later in the video, but first of all we're just going to do some general notes on what genetic engineering is. So basically it's the transfer of a gene that's desirable, so that codes for a particular protein that's useful. It's the transfer of that gene from one organism to another. That gene can be from a different species or it can be from the same species, but either way it would then change the genome of the new organism, and we would then call that new organism genetically modified. So the genome is the entire set of genes in one organism. So we'll change that genome and then it will result in that organism being genetically modified. And then that's GM for short. So if you see GM, that means genetically modified. It could be the same or different species. For example, this is done a lot with crops. So crops that are grown for food can have their genomes modified to have a gene, for example, that kills pests. So when insects, for example, try and feed on the crop, they get killed. It can also have genes for resistance to herbicides. So a farmer can spray an entire field with a herbicide. It would kill the weeds, but not the crops. So there's all sorts of different GM crops that are currently available. But also another example that we're going to look at today is when bacteria can be genetically modified to produce the human gene for insulin. So that's what we're going to look at now. So up here we've got a section of DNA from a human, and then that DNA can be removed and the gene for the insulin can be cut out of the DNA, and that is done using enzymes. So we're going to go step one up here. So step one, so the desirable insulin gene is removed using enzymes. And when that is done, the gene is actually left with what we call sticky ends. And those sticky ends are a short sequence of bases that are left exposed, so not double-stranded. I'll explain why that is later. So if this is the gene, let's do it in green as it is. Let's do it up here. So there's the gene. So that would be double-stranded. But then single strands are kind of left on either end. These are called sticky ends. They would have a few bases exposed that can then stick to the plasmid later on. So let's say there's 3 bases exposed here. Let's say, I don't know, that's ATT. This is CCG. And meanwhile, over here, we've got the bacteria. The bacteria contain rings of DNA called plasmids. These plasmids can be removed from bacteria. They can be cut open and have this desirable gene inserted into that plasmid. We call these plasmids vectors in this instance because they are able to be modified and then go back into the cell and modify the genome. So it can insert new DNA into the cell, into the bacteria cell. So the plasmid here is the vector. So step 2, a plasmid is removed from the bacteria, and another set of enzymes will cut the plasmid open. So plasmid is removed from bacteria and enzymes used to cut it open. So as before with the desirable gene, sticky ends are left on the ends here and here so that they can then be stuck or bind to these exposed genes, exposed bases here. So this will look something like this. If we just expand this, it will look something, you'll have to excuse my artistic skills. So we've got exposed bases on the plasmid that are going to be complementary to the exposed bases on the gene. So it would be T, A and A, which is going to bind to A, T and T. And on this end, we're going to have, just turn it round, G, G, C, which is going to be complementary to CCG here. So they will bind to those complementary bases. So plasmid is removed from bacteria and enzyme used to cut open. Sticky ends left that are complementary bases to the gene. The next step is here. So that desirable gene is combined with the plasmid. Those bases are, because they're complementary, they will join, they will form bonds and join. So the insulin gene will bind to the plasmid. So this plasmid has now been genetically modified. The final step is the plasmid that's modified is inserted back into the bacterium. And we now say that this bacteria has been genetically modified. It can go on and it will divide. It will produce the protein here. It will produce insulin, and then that insulin can just be harvested by the scientists. So the modified plasmid is inserted back into the bacterium. The bacterium is now genetically modified. It will go on to produce insulin. And that is an overview of how insulin is kind of produced en masse to meet our demands. I hope that helps.